Thank you very much, and thank you for bringing me in here.
Happy to have you all the way from, where are you, Norway?
No, I'm in Denmark these days.
You're in Denmark these days, okay. That'll be interesting. So please go ahead with your presentation and I will be back with a Q&A.
Excellent. Well, if we look at I'll just jump on the first slides very quickly, because that is the usual disclaimer, etc. And I'll start here. The molecule that we have at hand and which we have developed extensively is called BSG-005. This was actually coming out of some basic research going on in the technical university in Trondheim in Norway. We choose BSG-005 among about 20 to 25 different versions of this protein macrolide product called nystatin that we did some genetically formulation changes in order to get it improved compared to what was in this drug class originally. So it is basically a naturally occurring fungicidal chemical that is synthesized in the bacterial strain called Streptomyces norsai. And this is something we have been working on, and we have a good and solid patent portfolio behind the product. The key point here is that it is safe. It has, in all our toxicology programs, it had no kidney toxicity, no clinical relevant impact on liver, and also not on other blood parameters. It has been free of any cardiovascular, central nervous, and respiratory adverse effects in this toxicological development. The mechanism of action is the same as it is for this class of drugs. It means that the molecule in its formulated version binds to ergosterol, which is a fatty acid in the cell wall of fungi, and this leads to cell wall pores, and then ions and other matter can leak out, and that is causing cell death very efficiently. Ergosterol is fortunately not present in humans, but only in every fungal cell wall in those fungi that is attacking humans. And the difference here is that it's a fungicidal product and it kills the fungus. It belongs to the polyene macrolide group of products, and they are known to cause fungus death. And that is, in comparison to most other antifungal products, an improvement, or it's just better, because many of the existing products, they only inhibit the fungal growth. They don't kill the fungus. And it actually causes more than 1.7 million deaths a year, this disease, invasive fungal infections. And this is a life-threatening condition, disease, and it requires a fast diagnosis and an optimal management and treatment to try to avoid the high mortality and morbidity that you see in these diseases. As you can see from this graph, we have about 1.7 million, and this is more deaths than you see from many other well-known cancer types. And very often, patients who have this infection, they'd actually die from this one and not from the basic disease that they are being treated for. For instance, cancer. I'll come back to that a little bit later. And this situation is now seen by WHO, as well as the Center for Disease Control and Prevention in the US. Both of these organizations has realized this, and they had only a couple of weeks ago, weeks where they tried to inform the global and community about these issues. So what are these fungi diseases? What do they do and what are they? Well, basically, everyone that has a weak immune system, are in danger of getting such an infection. This is typically cancer patients, patients who have received an organ or hematological transplantation, people with HIV, patients with diabetes, and patients with severe lung diseases. They are at risk. And you can see on the right side of this one that if you look at it, those are the four main types of diseases that you get. It's candidiasis, it's called aspergillosis, cryptococcosis, and pneumocystis pneumonia, or Candida auris. And you can see that they are relative often seen in these patient categories. The other thing that is important is that there is a resistance development among the fungi. They are starting more and more to be able to resist the treatment with the standard products. And that means that we have an increasing number of resistant strains, and the number in general is causing alarm for the WHO and other places and other organs as well. And the reason is that these diseases have a very high mortality rate. Basically, it is anywhere between 20% and up to 90 or sometimes 95%, depending on what strain you are infected with and where you are in the world. BSG has what we call a unique profile when we test it against various kinds of fungals. So what we have done is we have tested it against the cells, and we have compared it with amphotericin B, which is the same class of drug as BSG 005, with a group called azoles, where we have tested most of also the newest ones from this type of drug, and of course the echinocandins, where we have seen that these three groups are the ones that are being used in the antifungal treatment for this disease, etc. What the slide here shows you is that everything that is green is good, everything that is yellow is questionable, and everything that is red is bad. And the only ones that has green all over the place for all the different types of fungi strains that we're looking at is BSG 005 and amphotericin B. Both of these products have a broad activity against various fungi strains. The only limiting factor for amphotericin B is that it has side effects. And I'll come back to that, but it's again a talk about kidney problems when you dose amphotericin B at higher doses or for longer time. But that is a key point here. So the takeaway message is BSG has a broad effect and it's safe, and all the other products have problems, either with side effects or not being able to handle every single strain of fungus that is involved. And exactly the drug resistance is growing. And that is why there is a strong need for BSG 005, also because amphotericin B has the side effects I mentioned earlier on. All this is giving rise to concerns, and it's being mentioned everywhere, and also by organizations like CDC and WHO. And if you don't have a safe drug and a broad efficacy, you have a problem in general. Just a little bit on the antifungal drug market. These data are a couple of years old, and I know that the market today is plus $20 billion US. And it is split between these different classes of drugs, the purines, the azoles, the echinocandins, and those that are treated with other products as well. The main one that we look at is the purine macrolide class, which is amphotericin B, candicicin, and nystatin. And if we look at the sales of this, it's about 10% of the market, or in today's numbers, it's approximately $2 billion US. The aim of BSG-005 is to become a first-line product, which is the treatment that you give for any invasive fungal infection. And if we can get that far at some point in time, then we will grow the polyene market significantly, and we will take market share from the azoles and the echinocandins. We have, from our phase 1 trial that we completed earlier this year, come out with some top-line results. And the main thing is it's safe. There was no significant side effects. And this was in a classical designed phase 1 study where we dosed first subjects with a single dose, and then we dosed subjects with multiple dosing for 7 days. And in this study and these two ways of treating the healthy subjects, BSG was found by our data committee to be safe in these healthy subjects. We had no serious adverse events, nothing at all. And those adverse events that we saw were all mild to moderate in severity. There were no clinically relevant changes in the laboratory parameters that we follow, and particularly not in the kidneys, nothing in the liver, and nothing on the potassium and other laboratory parameters that we were looking for. And we didn't find any relevant electrocardiograph changes, ECG changes, during this study. And what this means is that the data are confirming what we have, that the BSG is safe, and that we are ready to go into patients with an invasive fungal infection. I need to also, you haven't seen it advertised, that we have signed a co-development agreement with Alchem Laboratories of India. It's a large company with more than 17,000 employees. And it will give us, with this partnership, access to a lot of patients with severe invasive fungal diseases. It's because it's in India. Alchem will manage the trials, and they will fund all the trials we do in phase 2 and phase 3 for India. We will supply BSG-005 for these trials, and this will reduce our clinical development cost significantly. We estimate that if we let this go on with phase 2 and phase 3, it's an estimated financial burden of 300 to 400 million SEK that is taken away from Biosergen and its investors in this development. And having access to this high number of patients with invasive fungal diseases will probably reduce the development time to first market launch significantly. We can add centers and patients outside of India to all clinical trials, and they are all run according to FDA and EMA standards. We expect that about 70% or up to 70% of all patients in our development program will come from India. So our further clinical development is what we're working on right now. We will do a first study in patients, which we will expect to submit to the agencies in this week, hopefully. It will contain a phase 1b/2a with 15 and 30 patients, in total 45 patients, in a single arm, multi-center, open label dose escalation to assess the safety and efficacy of BSG-005 in patients with an invasive fungal infection where there are limited treatment options. And the aim is to get data that will give us proof of concept in patients with invasive fungal infections. We want to optimize the dose selection before we run into phase 2b, which is the prominent development phase that we have to get into. And we also hope that the data can give us a potential way to an expanded compassionate trial use program. The population we're focusing on in the first 30, 45 patients here are patients who cannot tolerate amphotericin B, typically because of kidney problems. We will look at patients who are azole resistant or has failed on first-line treatment. And we will look at patients with mild to moderate kidney impairment because they have limited options for treatment. And these are strongholds of what we see as advantages of BSG005 compared to other products in the market. So we expect from this trial to get a general safety and tolerability profile in patients. We see it also in patients that have no other treatment options. And then we will see it in different fungal diseases. That can be candidiasis, can be any of the others, aspergillosis, mucormycosis, etc. And we will get proof that we can cover the most difficult and resistant invasive fungal infections in this area. And again, we will see what patients can be handled with our product if they have moderate or mild kidney impairment and have really not many other options. This will risk mitigate our dose levels for the phase 2 patients and the phase 3 patients, and we will have an expanded use or compassionate use program initiation at hand as well. That's what we aim to go for. This is just to give you a quick overview on the timing of this. We have the first 15 patients, the second 30 patients. We have the timelines that is going in. We expect to submit here in 23, in hopefully this week. And then we will continue and start working on the next, if the data are positive. If the data is not coming out as we want them, we'll have to rethink what we're looking at. And then we have the phase 2B protocols coming up and potentially a compassionate use program also outside of India. On the manufacturing side, we have to resupply for the 30 patients that we will do. We also have to, for the phase 2B studies, to do some development work and make further manufacture to cover the needs of these trials. And nothing on the CMC. We basically need to manufacture for Alchem for the phase 2 trials. We have a small team. Management is myself and our CFO, Niels Laursen, and our COO, Tine Kolle Olesen. She's running all the clinical trial programs and is working very well with Alchem. So we are looking forward to get things going. I think this is my last slide, so that would be okay.
Thank you so much, Peter, for your presentation, and we will move on to the Q&A. I actually have one first question from a viewer. Let's start off with that one. It says, why is the application and patient start postponed several times in the phase 2 trial?
The reason for that is, of course, that when we engaged us with Alchem in India, we decided together with them to actually increase the number of sites. We originally only wanted 2 sites, but in our discussions, we agreed that we would probably need 4 sites to be efficiently enough and have the patients coming in fast. That has, of course, some implications in terms of having the protocol finalized and have all the other information required ready for submission. And that has simply taken a couple of months extra. And that's why it is. That's why I'm saying it finally goes in, and I accept that we have been having a delay of some months.
Thank you, Peter. And I believe we have some questions in the audience here.
Yeah, the absence of nephrotoxicity is, of course, the main issue here. When I look at your homepage, you have a picture there that shows some, even though much lower effect on the kidneys. I may misremember, but I think it's some effect on the tubuli. Is that correct? That's one question. The other is, do we know exactly why amphotericin is nephrotoxic so that we can compare the substances and see that that cannot happen with BSG 555? 005.
Well, thank you. Yeah, thank you very much. It's relevant questions very much, because the absence of kidney toxicity that we have seen in both the animal studies, the toxicology studies, and also in our phase 1 study in the healthy volunteers is a key point. What we have done is to look at how does it compare with amphotericin B when you look at the kidneys, and the way we have done that in that particular test you're talking about is to look at something called NAG, which is an enzyme that you can measure, and which is an indication of damage to the tubuli in the kidneys. There are two things on it. We can see that there is basically no effect on the kidneys. I know that the curve says there is some effect, but you see, we go up to, I think it's 10 milligram per kilogram with our drug, without any rise compared to amphotericin B. And the other thing I want to say is that in our phase 1 trial, we have also seen that we have not, I think it's about 2% of all the BSG is actually excreted through the kidneys. So the level of BSG is it's very low. And I don't see any reason to believe that we would have kidney toxicity at all. So that is, and I think we have proven that now, and we'll see it also in the patients that comes up now. So that was your question number 1. And the question number 2, please.
Yeah, and do we know exactly why amphotericin is toxic so that we can compare the structure of the substances and through that be more certain that we will indeed not cause any problems?
Well, the only issue here is that, and I think it's the main thing I already mentioned, we are not excreting BSG through the kidneys. The amount is so low, I think the risk is absolutely not existing. And that is the difference to everything I know between amphotericin B and BSG 005. And we have data that shows this from our phase 1. We did a lot of kinetic investigations, both on the plasma, but also on the kidneys, urine excretion. And the data are very convincing, as I see it.
And a question regarding the patents. Have we covered as many versions of BSG 005 so that no one can possibly come with a not identical substance, but close to and compete with us?
Well, I think we have one very, very good reason to believe that we are safe here. There has never been one potential, what should we say, intruder with a similar molecule. And I know it's because we had almost between 25 and 30 different versions. And many are exemplified in our patent. So I'm pretty sure we are on safe grounds.
Yeah, we have some more questions here from the audience. Please go ahead.
Yes, hello. So you will co-develop BSG 005 for the Indian market with Alchem. Can you say anything about any upfront payments or royalty rates or anything about this?
Yeah, I can tell you we have not agreed on any down payment on signing of such a co-development agreement. We have a low royalty that we will get once they hit the Indian market. And we'll get that overall. Where I believe we will see our income in this agreement is, of course, less financial burden to ourselves, but also that we deliver product to Alchem. And that is one of those ways that you normally earn quite a bit of money in this respect.
Okay, and with the new deal with Alchem, how will your cash burn rate change? Do you see how long you will last with the current money you have in the bank? When do you expect you need to either take in new money or find a new strategic partner, etc.?
Yeah, I can tell you that we have a reasonable burn rate now, and it'll only go down because, as you can see from the presentation and from what I've told you, the financial burden we have is much lower now. It means that basically we only need to produce and manufacture BSG-005 for Alchem. Plus, if we need to add some patients from outside of India to the global file. This means that we will raise money sometimes within the next 6 months and be able to run for 1 to 2 years with that. Because our cash burn is going dramatically down from next year.
Okay, and finally, when you do the first part of the upcoming study, will you be doing it with a Indian-based domestic CRO, or will Alchem sort you out, or how is that set up?
The setup is so that this is basically our study. We are the international sponsor of it. Alchem has an agreement with a preferred CRO in India, and they will run the study for them or together with them, and we will be part of that. So we have a three-party agreement on running this trial in India.
Okay, and it's safe to assume that a domestic Indian-based CRO, it will become cheaper per patient, I suppose?
That is correct. That is correct.
Okay, thank you.